Good morning, everyone. It's a pleasure to speak with you about the All of Us research program and talk about some of the work we've done to try to expand and create a national resource for science in this program that will eventually cover all life courses and life history to improve precision medicine for everyone. As we think about precision medicine, think about people having different disease risk. And so we're all familiar with people who come in and have maybe healthy, but eventually we all develop diseases. And those conditions can be experienced variably. We can have severe disease of a typical type. We could have self-limited disease. We think about something like a variable presentation of an infectious illness, certainly something we're all thinking about COVID-19 right now. And an atypical disease. Where you can think of something like lupus that can present in so many different ways. Similarly, people have variable drug response, and we all, of course, as we prescribe medications, seek to have the benefit. But sometimes the drug has no benefit, and sometimes the drug has side effects, and sometimes the drug only has side effects. And certainly, we would like to be able to use molecular mechanisms to target medications more precisely to individuals to obtain the benefit and reduce the toxicity. As we think about longitudinal cohorts and how we treat individuals and how we've made population health better, one of the most notable examples is the Framingham heart study, which began in 1948 and recruited about 5,000 individuals initially. Those individuals have contributed to so many findings. You can see here the first paper published from Framingham about 13 years later, where they found that people with cholesterols of 300 and systolic blood pressures of 180, had much higher risks of coronary heart disease. And those kinds of things were found to be interventable risk factors for coronary heart disease that resulted in future treatments to lower cholesterol, to lower blood pressure. And similarly, things like the importance of diet and exercise and smoking have been found through longitudinal cohorts, such as the Framingham heart study. And you can see with this slide from the CDC here on the right, that the mortality from cardiovascular disease has fallen dramatically over the ensuing decades, as we've been able to find particular treatments and had public health measures to improve cardiovascular health. Similar such curves have been found in things like cancer and other diseases, as we've better understood the diseases and created interventions that reduce our risk of disease and mortality from those diseases. And so our question is, can we have a similar experience with precision medicine in the next 40 years? And that's our question guiding this. And here's one example of this. So we're going with the same example of coronary heart disease. This example studied individuals who have residual coronary heart disease risk, despite being on class-leading medications for most of our populations, statins, which reduce cholesterol. And we found that individuals who had a subsequent myocardial infarction or revascularization event had genetic markers in LPA. And if you look at this target, we found that if we looked at a phenome-wide association study of this locus, that individuals with these variants were at increased risk of coronary heart disease. And importantly, all the different associations in that domain were typically related to cardiovascular disease. Fortunately, we did not identify other particular risks that would come up potentially with treatment of this. So this kind of analysis lets us look at potential indications for and side effects for a drug that would target this locus. And this is important because these individuals with genetic variants in this region had increased risk of cardiovascular disease, even when they had ideal LDL cholesterols of 70 or so. And in those individuals were even at increased risk compared to ones who had other elevated LDL cholesterols. So this is a model for how precision medicine may accelerate or help us think about particular drug treatments. And this sort of model can be extended to help us think about and accelerate discovery of the association of genetic variants for, in this point, HLA serotypes across different disease classes. And in this case, with just nearly 40,000 people, a large study compared to something like Framingham, but a small study compared to many of our modern day cohorts that can have 500,000 or more participants. We were able to see a number of associations of autoimmune diseases with these given HLA types. And you can see that individually, some of these different classes of HLA types are associated with more diseases than others. And it also helps us look at them in collectively as well as individually. In addition, many diseases have a phenotype pattern, and we can look at this to discover more about given diseases, and we believe maybe even find people who aren't diagnosed yet. So if you think about a disease like cystic fibrosis, which hits a number of different organ systems, it's composed of a number of different symptoms. So bronchiectasis, pancreatitis, asthma, failure to thrive are just some of these. But each of these has different impacts towards the disease. And something like asthma is much more common, and in this weighting scheme would not be worth as much as something like bronchiectasis. So you can add these together and understand one individual with these clusters of symptoms may have a higher risk or potential likelihood of having cystic fibrosis than another individual. And indeed, that is true, that if we look at patients who have cystic fibrosis versus those who don't, you can see elevated scores are found in those who have the disease versus those who don't. And you can distinguish these individuals without using the diagnosis of cystic fibrosis itself. And so that may help us think about severe, this individual later develop auditory neuropathy and blurry vision, eventually around age 34, muscle spasm and neuropathy, and because of weakness became confined to a wheelchair, and then developed dysphagia and needed a peg tube inserted, and eventually developed respiratory failure and needed intubation. This individual presented to the Undiagnosed Disease Network with a detailed history exam and whole genome sequencing, was diagnosed as a riboflavin deficiency, and supplementation with megadose riboflavin allowed her to regain much of her sensation and begin walking without a walker again, and certainly to be extubated. And so this is something where with a precision medicine approach, incorporating sequencing and molecular testing, resulted in actually a very cheap intervention that dramatically improved her life. And as we go back and look at time, we can see her phenotype risk score was dramatically elevated at 14 standard deviations, 14 times above what would be expected. And so that may help us think through how to process things like her genomic sequencing. One of the real problems, though, to understanding genomics, and really it has been a lack of diversity of those that we have genotyped and sequenced. You can see that the population that we've genotyped is dominated by those who are of European ancestry. And it's gotten a little bit more diverse from between 2009 and 2016. But even today, we see the vast majority of people who are sequenced or genotyped are of European ancestry. And if you look at the diversity of those who are not European, most of those represent people of Asian ancestry. And this bottom 4% represents about a third of the US population by demographics. And we know, of course, the US is becoming more diverse. So this represents a real opportunity for us to improve the diversity of the genotype samples. And this is the kind of thing that has impacts on everyone. And so here is just a few of a smattering of publications that have pointed this out. The importance of things like our clinical algorithms, clinical scores, and calculation of glomerular filtration rate are biased by the fact that we don't understand and really think about the true biological elements of race correction in it. And we can actually, incorporating some of those things can perpetuate systemic racism in our treatment of patients. Our polygenic risk scores, by not having diverse metrics in diverse populations who have been genotyped and sequenced, are not nearly as accurate for populations who are not of European ancestry. And you can see that here. And then when we interpret genetic variants, we impoverish our understanding of what's happening. And we call some genetic variants as being pathogenic, for instance, when they clearly are not, because and that conclusion was potentially arrived on because of the fact that we haven't sequenced a diverse enough population. And so as we extend the diversity of our samples, we improve healthcare for everyone, and also can help tackle some of health disparities. Much of the need for increased diversity and increased data on a population is some of the driving connections and reasons for all of us. And we hope that all of us will provide answers and help make connections across different populations. I include here two URLs for all of us: joinallofus.org, which is where participants can join and the program and get data around themselves as they contribute to the program, but also participate, answer survey questions, and schedule visits and that sort of thing. And then researchallofus.org, which is where researchers go and can access the data. The participant side of the program was launched nationally in 2018. We really value participants as partners in this program, and so we value to do things like give data back to participants and seek their input on things from our survey modules to how we return results. And they're involved in all areas of governance of the program. We've enrolled over 350,000 participants since we launched, and over 270,000 of those have contributed biospecimens. Importantly, over 50% of our population that are our core participants that have contributed biospecimens are of diverse race and ethnicities, and over 80% are underrepresented in biomedical research. And you can see that we collect a variety of different kinds of data types and individuals. They answer health surveys, they contribute biospecimens, electronic health records, physical measurements, and digital health technologies. And through these, we hope to really capture across the environment, lifestyle, and biological changes. So as we've collected a lot of data across this participant population, what we see is people enrolled in the program starting in 2017 as the beta phase of the program, and after our national launch in 2018. And they can contribute surveys from after that time. But one of the data types we've collected are electronic health records. And if you look at this slide here, you can see that individuals have longitudinal electronic health record data going back decades at times before they were enrolled. So this allows you to do these longitudinal kinds of studies long before the participant enrolled. And it also gives you quite a lot of data that you can analyze. You can see millions of conditions, millions of procedures that were diagnosed in individuals, and lots of laboratory measurements, drug exposures, that you can look at those exposures and the effects within the electronic health record, in addition to the survey data. And of course, these individuals also are contributing biospecimens. They have the option of contributing digital health technology data, such as Fitbits or other personal health activity monitors, and things like that over time. There are many ways in which we can use these data, and we've done a series of what we call demonstration projects for these participants. A number of researchers came in to help us test the system to replicate essentially known science. And now, since it's been open, of course, many other researchers are using the program. One of the things I wanted to highlight here was just one of these example projects. We looked at identifying participants who had been diagnosed with depression, and we looked at their medication usage by class. And here you can see that the most common medications used in white individuals compared to those who are non-white were SSRIs, which is probably not surprising to those who have treated patients with depression. But you can look at the second, third, fourth, fifth, sixth medications here and notice there's some significant differences, and this is statistically significant. Of course, we'd like to do a lot more with this kind of example, but and drive into differences that may vary by region of the country, to certainly break down into the categories into more precise ones as well, and people give us a lot more information about who they are and other components. But here we are just trying to, with this initial study, provide a blueprint for other people as a springboard of what kind of analyses they can do. And so as we think about all these data sets coming together, all of us is just a part of the international efforts to pull data together across diverse resources and make them interoperable. Because the paradox of personalized medicine is we really need huge populations to better understand how to care for a single person. And that gets to understanding the rare genetic variants we talked about before, to understanding differences in clinical trials. And what we're working to do since some of these national and global efforts are to figure out how we get the different data sets to work together and create common protocols, common application programming interfaces, and data models, such that data can be shared and protocols developed such that we can work together to better answer these questions across the lifespan.